structure of the DNA, it's the double helix, and it consists of 2 strands, which is why it's called the double helix, and it twists together to form the actual DNA strand, and it coils. And it consists of 4 nitrogenous bases, which allow DNA replication and protein synthesis to happen, and they are cytosine, adenine, guanine, and thymine, and they are complementary base pairs, and I'll open it so you can see. And cytosine and guanine go together every time, and thymine and adenine go together every time. And they are also nucleotides, which are the individual actual little bases of A, T, C, and G. And they are also divided into purines, which is adenine and guanine, and pyrimidines, which are thymine and cytosine. And also hydrogen bonds hold the actual complementary base pairs together, which keep them from falling apart. It's these lines right here. And the sugar phosphate backbone is what holds the actual structure together. It's located at the sides, and it allows it to coil without falling apart. And the genetic code, which is like actual DNA, which comes from the DNA that is like kept in the center of it, it decides what traits we have and what we look like. Application, you have the DNA polymerase, which is along the Okazaki fragments, which are created, but I'll talk about those in a second. It's responsible for DNA replication and needs an RNA primer. And the RNA primer is a single strand of RNA that acts as the starting point of DNA synthesis, and it goes along the insides of the part that's torn apart. And the helicase is what unwinds the DNA double helix in replication, and so it's what actually takes it apart so the RNA can go along the inside. And the replication fork is this area down at the bottom where it's the area where the double helix has unwinded and the DNA will be replicated. And the leading strand is the one where DNA synthesis occurs continuously towards the replication fork. And the lagging strand is where synthesis restarts many times as the helix unwinds and causes short fragments of nucleotides called Okazaki fragments. And this will be the lagging strand. And we'll say that like this is an Okazaki fragment. And so the DNA polymerase is located in the Okazaki fragment. And ligase, which is what binds the Okazaki fragments together into one DNA molecule by sealing the gaps between them. And so it creates this line down the center where it's sealed together, and it creates a double helix where the bond is done and it can curve and it won't fall apart. For transcription, you have mRNA, which are messenger RNA that carry the coding sequences for protein synthesis, and this is the mRNA strand, the top one. And you have the RNA polymerase, which is this big thing, and it separates the DNA strands and hooks together the RNA nucleotides, which allow transcription to happen. And so for translation, the tRNA, or transfer RNA, translates the mRNA into amino acids, and so as the original strand of DNA gets pulled through the RNA polymerase, it turns it into mRNA. And the tRNA turns the mRNA codons, which are 3 of the mRNA, it turns it into amino acids, and that is like, it's the 3 bases, which is the codon, and the amino acids regulate the translation of the RNA so that it's not happening too fast or too slow, and so that there's not deletion or insertion, which causes point mutation and which causes nonsense, because when the DNA is not able to be read properly due to insertion or deletion, that's nonsense. And point mutations can also occur when this happens, and it's alterations in the placement or structure of a gene that can be caused by damage to the DNA, chemicals, many things.